Hi guys! Well, we are on passive transport with the topic of the plasma membrane. So this learning objective is going to cover describe selective permeability. And before we get into talking about selective permeability, we just want to first sort of orient you of where this is happening. So this is happening with the plasma membrane. So if you're looking here at the cell, the plasma membrane is going to be the part that is surrounding the cell. So here we have the plasma membrane where I'm highlighting it here in this black, and this black here surrounding the cell. This is going to be our plasma membrane. Okay, so that's just one way I wanted to orient you. The other thing I wanted you to know is when I say the plasma membrane, sometimes also in the text, when you hear the word a cell membrane, they mean the same thing. So the plasma membrane, also known as a cell membrane. So I just wanted to make sure I point that out before we move forward. Okay. So to start this discussion, what is it? What is it and why? So why is a plasma membrane important? Well, we have a couple of reasons here. The plasma membrane is needed to form a cell. Without the plasma membrane, we won't generally have a cell. The other reason is that it helps maintain a cellular internal environment, and that is homeostasis. So let's go ahead and add that. So it helps maintain a cellular internal So it helps maintain a cellular internal environment, which is homeostasis. So it helps maintain an area inside the cell here, which I'm kind of coloring here in yellow, keeps it at a nice homeostasis level so that all the parts can work correctly inside the cell. So that's what it helps do. So when we're talking about the plasma membrane, we have to highlight what its function is. So what is the function of the plasma membrane? Well, it says the plasma membrane separates the internal environment of the cell from its external environment, and it does so by controlling what molecules enter or exit the cell. So the plasma membrane separates the internal environment of the cell from its external environment. It does so by controlling what molecules enter or exit the cell. So it's going to control what comes into the cell, what comes out of the cell. That's the function of the plasma membrane. So what's interesting about the plasma membrane, now here we highlighted in like a really dark line, but then if you actually look at what makes it up, it's made up of a lot of parts. So for one, it's made up of these phospholipids, of glycoproteins, peripheral proteins, cholesterol, integral proteins, extracellular matrix, glycolipids, cytoskeletal filaments. Try saying that all really fast. Okay, but it's made up of all these parts. And what's interesting about these parts is that when it comes together, these parts are all known as this fluid mosaic. So these parts all make up this fluid mosaic model of the membrane structure. So one way you can think about this is, let's say you're looking at a mosaic, and this mosaic is, I've seen a couple, I went to Santa Fe recently, and there was a couple walls there in Santa Fe where there's a mosaic, which is really several different tiles that have been broken up. They're different colors, and they're pieced together to make a picture, to make some image out of these tiles just by the different colors. Well, if you can take this plasma membrane and slap it up there on a wall, it has a fluid pattern. What's interesting about this pattern, though, and what makes it different than, let's say, that mosaic art piece at Santa Fe, is that this is fluid. So if I were to take my hand and stick it through a cell membrane, you would be able to just stick it right through, because it's made up of these phospholipids and these different proteins that allow it to sort of move out of space. So we can kind of like stick our hand through the wall. It would be really soft and sort of like jelly-like. And you can imagine why that's important. Why would cells need to have this ability to sort of change its shape in our body? So if we're thinking about, let's say, red blood cells have sort of like a rounded shape. And if you can imagine your red blood cells, or blood, being pushed through from the fingertips up through your shoulders, and it needs to kind of make this 90-degree angle, it's able to kind of squish itself into going past that corner in order to fit. And then it sort of just bounces back out. So it being able to be fluid could help it easily travel through the red bloodstream. There are some diseases that have an effect on that, especially on red blood cells, one of which is sickle cell disease. The sickle cell disease, their shape is rigid and also kind of spiky. And what it does is it sort of creates like a clump, or when it's trying to get through corners, it's not able to sort of just bleb into one and squish past one another. And that's what, and it has that effect on that cell structure. So just keeping in mind that this fluid mosaic model helps in having your cells being able to move. Another thing I want to point out when we're talking about the plasma membrane is that it is made up of a phospholipid bilayer. So the phospholipid bilayer is where we have a phospholipid, like one phospholipid combined with another phospholipid, and together it's two. So if you're thinking about the word bi, that is related to two. No, two layers. So this is two layers of phospholipid bilayer. And when you have this oriented in this direction, it is made up of a hydrophilic head. So philic being it likes water. So hydro being water, so it likes water. And then the other part, it likes water. And then what we have in the center here is this hydrophobic tail, which doesn't like water. So if we're thinking about it in that fashion, the head that do like water, they're pointing outward, and the areas that don't like water are going to be pointed inward. So that's what it uses when it comes to controlling what goes in and out of those parts. So now we get to talk about selective permeability. So selective permeability is where some molecules freely pass through the membrane, or they need a way of passage. So those molecules that can easily pass through the membrane are going to be those that are small and uncharged. So small and uncharged molecules can pass through the membrane. They can freely go and sneak their way through or past the phosphophilic heads, past the phosphobic tails, or hydrophobic tails, and then the phospho in the hydrophilic head. Oh, I don't know why my brain's going back and forth. So what I mean by that is if it's small and uncharged, let me go back to this part here. If it's small and uncharged, it's going to be able to just make its way through this phospholipid membrane to the inside and the outside because they're able to sort of shimmy their way between the phosphophilic head and the hydrophobic tail. It's until they are large molecules that they become a little bit hard to use, or not hard to use, but they have trouble getting through. So molecules that don't freely pass through the cell membrane are those that are large and those that are charged. Large molecules being those that are polymers. Polymers meaning they're many of something. So if a polysaccharide or a molecule that is, let's say, a polymer of nucleotides, those things are large. And then molecules that have difficulty passing through the phospholipid membrane are going to be those that are charged. So several of those are going to be ions that have a plus charge or a negative charge. They have a positive charge or negative charge. So those ones are the ones that have difficulty in passing through the membrane. So then when we're talking about passing through the membrane, we generally have to ask ourselves this question, which direction are they moving? So which direction do these molecules move? How do they know whether they are going to go into the cell, or how do they know they're going to go outside of the cell? So here's where we're going to be talking about diffusion. So diffusion is a passive free movement of a small uncharged molecule across the membrane towards equilibrium. And I'll say that again. Diffusion is passive free movement of a small uncharged molecules across the membrane towards equilibrium. So they're moving. Here it says uncharged, but in this case, things can also move that are charged, and it just depends on the gradient. So generally with diffusion, these are where molecules move from high concentration to a low concentration, or to a lower concentration. So that's usually the definition for diffusion. And I'm just going to point it out here because sometimes students confuse this with osmosis. Well, osmosis is still involving diffusion, but osmosis is the movement of water molecules from a high concentration to a low concentration. So here I want to just summarize what we chatted about before I bring up the activity. So if we go back to what we had started our conversation about, we were talking about the plasma membrane and why it was important. And then we went into the function of the plasma membrane, the main point being that it controls what goes in and out of the cell. That would be the plasma membrane. Then there's these different parts here, the components of the plasma membrane that make up this fluid mosaic model. And the main point out of that I wanted to point out was this formation of a phospholipid bilayer. Okay, so that's going in or out of the cell. Now we got into what is selectively permeable. And that's related to molecules moving through the plasma membrane. So molecules that freely move through the plasma membrane, those are small and uncharged. So these are going to be like gases. And molecules that don't freely pass through the cell membrane, these are going to be large molecules like polymers and charged molecules that have a plus or negative charge. And if they are both large and charged, they definitely can't just cruise through that plasma membrane. They would need some sort of channel protein or another type of protein to help it get through. And then how are molecules, like which directions are molecules moving? Well, that depends on diffusion. So diffusion is moving from a high concentration to low concentration. And then osmosis is the movement of water from a high concentration to low concentration. So those are the difference between those two. So here in the activity, I have it posted in the same module area there. And this activity, I want you to try this out. So what it's doing is it's starting off by giving a description of the plasma membrane. Now here looks like that fluid mosaic model. And then it also looks like it's not looks like, but also here it's talking about the hydrophilic head and the hydrophobic tail. But it gets to a point down here with these questions here, part A, it's asking you, will the following molecules be able to pass through the plasma membrane? Why or why not? So this question is going back to the part where small and uncharged molecules can pass through, and large and charged molecules cannot pass through. Okay, so we want to go back and review that. So in this case, oxygen, this O2 is a gas, and oxygen is uncharged, and it is a gas, so it'll be able to pass through. Then on the second part, we're talking about diffusion, moving from a higher concentration to a low concentration. And then the third part, which actually is one of my favorite types of problems, is that you're working on answering this question. So there's a concentration of molecules is shown both outside the cell and inside. Based on the chemical nature of the molecule shown, what would happen to each molecule? Would it enter, exit, or not pass through with diffusion only? And explain your answer for each. So here, you want to pay attention to what the question is asking you. So this question is specifically thinking about diffusion only. Diffusion only. So the first thing you want to do is to ask yourself, okay, here we have a sodium cation, so a sodium ion, and this is a charged molecule. Will it be able to just pass through the plasma membrane? And in this case, the plasma membrane is represented by this blue line. So that's going to be the plasma membrane. Let me go ahead and just put that in here. So is it going to pass through? Well, in this case, it's not. It's not going to pass through. So no, Na plus will not pass through plasma membrane because it is a charged ion. So it has a charge. So this one wouldn't pass through. But if it did, or it was able to move through, pay attention to these numbers here. There's some numbers here next to the ions, and these are units. So these numbers will help you indicate if it can pass through, which direction will it go through. And remember, that's back to diffusion. Okay? And then the last thing with this question down here, what would be the level of CO2 at equilibrium? So what equilibrium means is like what number would the molecule balance out on each side. So that's what it's getting at. All right, so I'm going to go ahead and let you guys try this out. Think about it, work on it, and then following this, I'm going to post the key to these problems.